Good morning and welcome to day 2 of the San Joaquin River Restoration Program's 2020 virtual science meeting. Thank you for joining us. We're very excited to have you here. My name is Josh Newcom and I handle public affairs for the program. We had a very eventful first day yesterday and I hope you appreciated our panels and expert speakers. I also hope you had an opportunity to visit our virtual open house poster session and talk with some of the authors that were there. There you'll be able to access the event agenda, which is also available in your documents drop-down menu. See the full bios of all of our presenters and view the science poster gallery, which is excellent. A few more reminders. Please remember that all attendee microphones have been muted and use of your webcam will be unavailable during the GoToWebinar portion of the meeting. We are on a pretty tight schedule. So if there is time, we will have question and answer opportunities today following each speaker's presentation and at the end of all the pop-up talks during that session. You can ask questions in 2 different ways. The preferred method is to submit questions at any time using the questions tab in your webinar dashboard control panel. It'll be relayed to our moderators and the moderators will read your question in turn. Or you may use the raise hand feature that you see in your webinar dashboard, and we'll do our best to track these. But again, the preferred method is to enter your questions through the chat feature. Questions will be responded to on a first come, first served basis. Staff will unmute your microphone when we are replying to your question. And if we don't have time to respond to your question due to constraints, you'll receive a reply from the appropriate speaker after the conference. There is a chat feature in the GoToWebinar, but we're asking attendees to please reserve that feature for relaying messages to the support team if you need to. Also, please note that we're recording this conference and plan to post a video version of this conference for those that weren't able to attend after it's finished. We'll announce the availability of these videos once they're posted. And finally, a quick note for our speakers. Please mute your microphone until you are presenting or responding to a question. Unless you are presenting or responding to a question, please keep your webcam off. And with that, I'd like to introduce our first speaker. John Carlos Garza is a supervisory research geneticist in the Fisheries Ecology Division of NOAA's Southwest Fisheries Science Center and an adjunct professor in the Department of Ocean Sciences at the University of California, Santa Cruz. Carlos leads the Molecular Ecology and Genetic Analysis Team, a joint NOAA-UCSC group that develops and uses molecular genetic data and analytic methods to address questions related to ecology, evolution, conservation, and management of marine and aquatic animals and ecosystems. Carlos received a BA and MS degrees from the University of California, San Diego, and a PhD from the University of California, Berkeley. He has 30 years experience in basic research and in the application of genetic methods to conservation and management issues. He has served on multiple federal technical recovery and biological review teams for salmon and trout, and on the scientific and statistical committee of the Pacific Fishery Management Council, as well as on the California Hatchery Scientific Review Group. And with that, I'd like to turn the screen over to Dr. Garza.
Good morning. Can you all hear me?
Yes, we can.
Josh? Okay, you can. Okay, great. Now I just need to figure out how to get back to my presentation and not my face when I'm talking. Let's see here. Okay. All right. Can you see the presentation now?
No, currently we just see you, Carlos.
Okay.
We were seeing your screen before.
All right, let's see here. Show. There we go. Oh, as soon as I talk, it takes me back to the, let's see here.
Carlos, in the GoToWebinar control panel, there should be a tab that says sharing. If you drop that down, there should be an option that says show screen. And if you click the screen menu, it gives you a drop down and it should let you select which screen you want to show or which individual window.
Right. Let's see here. That's what I was doing. Okay, just a window. Okay, let's see here. How about?
Yep, there you go.
Okay, it might switch back, but we'll see. Okay, great. So, sorry about that little technical glitch, and good morning and greetings from somewhere out here in the virtual universe we're all living in now. So today I'm going to tell you a little bit about the genetics work that has gone into the San Joaquin River Restoration Program. My co-author Anthony Clemento and I have been involved with the program since 2009, and I think that predates probably most of the people in this meeting. And so I'm going to give you a high-level overview of the various and sundry ways that genetics have and will continue to contribute to the successful reintroduction of salmon and perhaps eventually steelhead and sturgeon into the San Joaquin River restoration area. So I'm going to start by giving you just the briefest of overviews, since this is perhaps a group that does not need this overview at all. But just to keep this in context, so the Central Valley and the Sacramento-San Joaquin River system comprises just about a third of the land area of California. So it's the major geographic feature of our state. And it also is notable because it contains the southernmost populations of Chinook salmon, at least in their native habitat, as well as the most diversity in ecotypic identity, and particularly that related to migration timing. As you all know, historically there were a very large number of fish. Historical estimates are hard to come by, or accurate ones, but there were undoubtedly more than 1 million fish in some years. And the reason I put up this sort of famous fade to black slide here is just to re-emphasize how inordinately the construction of dams and water operations have affected access to the habitat, the upstream habitat of early migrating fish that would typically use these upriver habitats for summer holding and then often spawning in the fall. I think you probably all know this about Central Valley Spring Run Chinook, ESU, and its listing status, threatened as of 1999, and not doing better then or now than it was then. But the reason I'm showing you this is really more to tell you a little bit about where there are still spring Chinook in the Central Valley, and the primary locations where they're present and have been persistently present are obviously the Feather River, and then Butte Mill and Deer Creek. More recently, there have been reestablished populations that have been documented in a couple of places, including Battle Creek and Clear Creek programs that the U.S. Fish and Wildlife Service has been very involved in. So when things first started in the San Joaquin program, we had to figure out, what we had in terms of fish and what the appropriate donor stock might be for reintroduction, and that was the first order of business. So we used the techniques of phylogeography, using genetic markers to understand relationships and origins of different fish populations and different individual fish to try to understand the lay of the land, if you will, with respect to spring Chinook and their ancestry and relatedness to each other. And what I want you to note here is a couple of things. One is that the naturally spawning spring run, or the populations that have only naturally spawning spring run are all monophyletic. They're all each other's closest relatives. And this tree might make it look like they're actually derived from winter run, but it's actually the other way around. These trees can be hard to interpret, even for geneticists. And the second thing I want to show you is that I want you to notice from this is that the Feather River spring stock is in fact very much closely related to the Feather River fall stock, and in fact, we now know that that is primarily it's a true spring run stock, but it's one that is on a fall run, an ancestral fall run genetic background from presumably from the ancestral Feather River fall run, and some spring run fish that were crossed onto that background and then selected for to maintain that stock. So it seems like it would just be as easy as grabbing some fish and putting them in the restoration area and being done with it, but actually it was way more complicated than that, trying to figure out where to get fish. At least conceptually it was. And part of the reason for that is you really can't predict whether different groups of fish will or how they'll respond to newly available habitat. And often you just need to let fish sort of guide the way by, if possible, experimentally introducing multiple stocks and then carefully monitoring to see which ones do best. That said, and I should say we didn't have an obvious donor stock for the San Joaquin on the basis of geography, because everything in that part of the Central Valley had been extirpated. And as I told you, the Feather River is kind of a unique beast, even though it is geographically the most proximate of the persistent populations. But one of the guiding principles of the program, one that we're still working with today, is that the donor stock should, the broodstock selected for the San Joaquin should encompass the entire genetic and phenotypic diversity of the donor stock, which means, just as we can measure it with genetic markers and also things like the full age and life history complement in that donor stock. And very importantly, the process of selecting a donor stock need to make sure that it doesn't engender demographic risk to the donor stock itself. And as you might imagine, that made it the case that that mill and deer in particular have been off limits pretty much the entire time. Butte Creek has gone up and down in abundance over the time that the restoration program has been in action, and may be available at some point for broodstock mining. And as a consequence, both of the fragile state of the potential donor stocks that are naturally spawning, and the fact that the river is even now not fully online, a decision was made to use sort of relatively low value fish in the beginning of the program. And I just want to point out, as many of us have discussed, that is, so as you all, I think already know, what that meant was taking fish from the Feather River for the initial broodstock collection from the hatchery program there. But that's also somewhat contradictory with the goal of the technical advisory committee that was convened at the beginning of this program, and ongoing opinion in the program about not wanting the population that's building in the San Joaquin ultimately to be hatchery supported. Nevertheless, we did need to do a genetic evaluation to see whether what the technical advisory committee was recommending initially, which was Butte Creek as the most appropriate donor stock, whether the genetic data actually back that up. And in a nutshell, and I'm not going to go into this other than to tell you that it's still a pretty accurate compilation and comprehensive compilation of levels of genetic diversity and of the reasons why we believe that they are what they are for spring Chinook stocks in the Central Valley. And just in a nutshell, what we found is that the Feather River is genetically the most diverse of the spring run stocks by at least a small margin, but that may be at least partially because it's had a contribution from, or is maybe even primarily at the genomic level, a fall run stock. Conversely, Butte Creek is the least diverse of the stocks, even though numerically it has been the most abundant in a number of years. And the reason for that is quite clearly that there was a big bottleneck in the 1980s at the same time that the winter run almost disappeared. Butte Creek spring run also almost disappeared, and just a few hundred animals made it through that bottleneck, and they are the progenitors of the entire stock today, with the exception of perhaps a few migrants that entered the restoration area. Sorry, not the restoration area, Butte Creek, in the ensuing period. And it's worth pointing out that the Butte Creek spring run Chinook stock is the second least diverse Chinook salmon stock in the range of Chinook salmon that we have examined with a number of other collaborators. So with that in mind, that gives you some idea of how we got to where we are. But let me tell you now about what we're doing on an annual basis to evaluate and to support the program. So we work with the Department of Fish and Wildlife and the other collaborators every year to both confirm the stock and determine the genetic sex of the individuals that are brought from the Feather River hatchery as contributors to the San Joaquin program. We have a genetic sex assay that tells us that, and that's really important because the hatchery staff at the SCARF need to segregate males and females and change their feeding regimes to reduce precocious maturation. Stock ID has been radically improved in the recent past through work that we have been doing that has that we've done that has developed markers that are unambiguous identifiers of spring Chinook in the Central Valley. So we can, even in the Feather River, where using traditional techniques we would identify those fish as fall run, we can identify them unambiguously as spring run when they are. And this is the second one is a really something that we didn't foresee being as much of an issue as it has turned out to be. But there are a lot of related individuals, even in big stocks like the Feather River Spring Chinook stock, and they are they raise the risk of inbreeding, and it's not a trivial risk. I'll tell you more about that. And we've also been using the genetic markers and stock ID to identify and characterize all the fish that are volitionally entering the San Joaquin River. And in addition to that, any fish that does enter the San Joaquin River, either volitionally or through human transport, is getting a genotype, which will then genetically tag all of its progeny and allow them to be identified at any point in their life history, including down in the Delta or in ocean fisheries. And then eventually we hope to actually very carefully evaluate the donor stock, multi-stock donor stock strategy that is still on the books as part of the program when it's feasible to take fish from the other spring run populations in the valley. I think you all know this, but starting in 2012, Feather River Hatchery spring run egg lots began to be subdivided with a fraction of each of those egg lots a small fraction transferred to the scarf for rearing, and then eventually as broodstock when they mature. Anthony, as part of his dissertation, actually began doing a long-term pedigree analysis of the Feather River spring Chinook back in 2006. So we have full pedigree information for all of the fish in that program that are not natural origin and therefore have parents in the program from before. Just checking my time here. So what that allows us to do is identify the sibling relationships among the Feather River spawners and use that information to evaluate the effective population size, really sort of the true number of ancestors or founders to the San Joaquin River broodstock, and then use that information to decrease the probability of inbreeding. We know from a number of relatively definitive experiments that matings of brothers and sisters in salmon and steelhead results in dramatically decreased survival of their progeny. So it's something that we need to pay careful attention to. So just to give you some idea of what we're dealing with here, these are some pedigrees from the from the Feather River spring Chinook stock that is the source currently for the San Joaquin broodstock. And this bottom pedigree is one that includes almost 2,000 individuals, and actually would go, probably, into the next room for most of you if the screen continued that long. So just to be clear, that does not mean all 1,788 of those individuals are related, but they're all connected in a pedigree. More specifically, let's see here. Okay, why is that not changing? Oh, there we go. However, there are a lot of full siblings here, and what I'm showing you here is a graph over time of the number of offspring that a pair of Feather River spring Chinook return to the hatchery that are then selected as broodstock and used in the next generation. And two things to note here is that in some instances, over 20 of the broodstock in any given year are full siblings. Sorry, over 20 of the broodstock from a particular parent will be used as broodstock in some future year. And the other is that only about 1/3 of all of the broodstock at the Feather River hatchery in the spring program are unrelated to other broodstock in that program, unrelated at the level of full sibling. So there are a lot of siblings out there. And how does that play out in the San Joaquin? Well, it plays out quite substantially. This is a graph of or a table of the number of siblings or groups of size 2. That means there are 2 full siblings amongst the founders of the broodstock in the San Joaquin program, or in the case of sib ships of size 3, there are 3 full siblings, and up to 13 full siblings that contribute to the fertilized eggs that are brought to the San Joaquin as broodstock and program founders in some years. And as many as 2/3 of all of the individuals that contribute to the egg lots, the fertilized egg lots that are used as the source, have full siblings that also contribute. So you can imagine there are a lot of cousins and a lot of siblings in our broodstock program in the San Joaquin.
Hey, Carlos,
however, I do want to.
Sorry to interrupt,
yeah.
Carlos. We have about 1 minute left.
Okay.
So, sorry.
That's okay. However, we are maintaining heterozygosity quite substantially, so we're doing pretty well in that sense. Okay, I have 4 minutes on my clock, but maybe we start a little early. Okay, so I'm just going to skip over this technical information and just talk about the two most important things here. So one of the things that we do on an annual basis to minimize the inbreeding that comes from all of those siblings in the broodstock is the construction of what we typically call a mating matrix, which is simply a way of categorizing the relatedness of every potential male mate for every female, and using that information to avoid mating her with her close relatives, which are the individuals in red in each of these columns. Each of the columns is a female and all of her potential mates ranked by how closely related they are to her. So that dramatically reduces inbreeding and other negative effects of inbreeding in the broodstock. So this is my last data slide here. So the other big thing that we've been doing recently is the genetic analysis of downstream migrants. We're just starting to work on the fish from this year that were captured this year now. We've completed 2 years of analysis of the downstream migrant juveniles. And the goals for that are to understand the efficacy of the novel adult release, mature adult release program that has been used in the restoration area to identify progeny of natural returning adults that are brought into the restoration area, and also to estimate the relative reproductive success of individuals from different release strategies and different categories of fish. However, one of the more interesting things about the rotary screw trap analysis has been all the surprises that we've found, and everyone likes surprises, as do we. And the 3 that I want to point out to you here, and I can talk with you all about the details later at some point, is in both years we found progeny of scarf broodstock that were not known to have been released. We think that those are either trap efficiency fish that were not recognized as such, or unintended releases. In 2018, we discovered progeny of fish that were not documented in the restoration area. We're not sure how those fish got into the restoration area and spawned, but they spawned with some of the adult release fish and by themselves. And they were all spring Chinook, pretty interesting. And then this last year, 31 of the fish that were that we evaluated with the rotary screw trap samples were in fact not Chinook salmon. And when we did a species ID assay on them, we found that they were Oncorhynchus nurca, aka sockeye salmon or kokanee. And we were surprised to learn, and I felt ignorant that I didn't know this, that in fact kokanee are raised at the San Joaquin hatchery, and presumably those were unintended releases from the hatchery that were then caught in the rotary screw traps. So lots of interesting things. And going forward, you guys know all this stuff. It's basically most of things that I've just said. Adaptive management backed by strong monitoring, evaluation, and oversight is really what's going to be needed to determine success of the program. So lots of people to thank, and including you all for your attention.
Hey,
thanks.
Thank you, Carlos. Appreciate it. I'm sorry that we don't have time for any questions right now, and do need to keep to our schedule. But thank you for the great information.